Hello, everyone. Hopefully you've made it here to the stage. A very, very, very warm welcome to our webinar today. I'm Paula Burton, CEO and co-founder at Mass Dynamics. We're absolutely thrilled to have everyone here across multiple time zones in silico, if you like, and we do hope that wherever you are staying well, safe, and coping through this incredibly tricky time. So we're here presenting as part of the CASSS Mass Spec pre-conference webinar series, and we absolutely thank them for their support. It's likely that you're here because you'd like to learn more about what we do and how we do it. And so regardless of your role in the wonderful world of mass spectrometry, we've designed this webinar with the aim to have something useful for everyone. So let's get started with walking you through how we can support each of you and life scientists around the world to better transform mass spectrometry data to knowledge better, faster, and easier, and without having to make any compromises. We've got a jam-packed agenda today, and this is how we're going to work through it. So first up, we've got Andrew Webb. Now, Andrew is an associate professor at the Walter and Eliza Hall Institute, one of the founders of Mass Dynamics and our chief scientist. He's going to be sharing what he's learned across his mass spec journey and talk us through why Mass Dynamics exists, our approach, and the core principles that Mass Dynamics is based upon. We're then going to dive into each of the proteomics applications we offer. So we've got Joseph Bloom, one of our talented data scientists, and he's going to walk us through our discovery service and showcase how we deliver differential expression analysis. And then Giuseppe Infusini, one of our co-founders, excuse me, and our proteomics informatics leads, will take us through product characterizations to services, peptide mapping, and quality attribute monitoring. We'll then talk you through where we're heading as a company and share with you a very special announcement. We'll close the formal part of the presentation and open up live Q&A. And we're aiming to have the presentation part of the session complete in around 40 minutes or so. So there's going to be plenty of time for discussion and Q&A afterwards. So some housekeeping before we start. We will be recording this session, and this will include any questions that come through the discussion. If you've got any questions throughout the presentation, please submit them in the Q&A area of the Hop-In platform so we can address them at the end. I'll now hand over to Andrew Webb to kick us off. Thanks, Andrew.
Thanks, Paula. So from my perspective, it's an incredibly exciting time to be a scientist in our field. It feels that the value of mass spectrometry that it's bringing to life science is really beginning to be truly appreciated, and that it's making substantial contributions in more and more disciplines, something I strongly believe that is set to continue for the future. I think the easiest way to explain what Mass Dynamics is explain to where it originated. For the past 10 years, I've had the privilege to lead proteomics research at the Walter and Eliza Hall Institute in Melbourne, Australia. And over this time, we've worked with hundreds of scientists on a wide range of projects, which has given me the opportunity to identify and study the major challenges and bottlenecks that occur when we work across large and often highly multidisciplinary teams to provide research outcomes. It's these challenges that Mass Dynamics was created to solve. Since beginning Mass Dynamics, I've been able to speak to many peers across the range of industries that utilize mass spectrometry and have been surprised to learn about the commonality of these challenges across industries, from academia to food manufacturers and in biotech, both large and small. At a high level, all experiments share a common flow where we start with a question and then acquire data. When we process this data, we assess quality and we aim to create insights that ultimately do something with this new knowledge. This simple figure here reflects the simplicity of the flow in concept, but in practice, as I'm sure you're all familiar with, it's anything but simple. The trouble is that at a fundamental level, many of the steps involved are driven by different groups of individuals, sometimes with very different backgrounds, skill sets, and even scientific languages that are required to work together to achieve common goals. Add to this the disjointed and disparate software tools that many of the multidisciplinary teams have to use to complete their jobs. These include handoff points and many other manual steps as well. This, as I've personally experienced, can routinely blow out relatively simple research projects into many months of work for a single experiment. And we haven't even gotten to the most crucial part of the process, which is where we want to try and create knowledge from our experiments. Something that I think the entire field has really struggled with as data sets have ballooned and grown of late. To summarize, through our insights, we've been able to identify the major bottlenecks preventing us from iterating and moving at pace towards breakthrough discoveries and developments utilizing mass spectrometry. We've heard so many anecdotes along our journey, such as, have I missed something in the results? Or I feel frustrated and underutilized. And one of my all-time favorite quotes was from a director at a leading biopharma, where he stated, I can't tell you how long this part takes because often we don't get that far when it comes to some of the later analysis points of some data sets. From the perspective of mass dynamics, we see these challenges as opportunities to dramatically improve the speed of processing, opportunities to reduce dependencies on software expertise, and increase efficiencies in sharing and collaborating on complex data sets. Ultimately, it's the speed and depth of knowledge gained from this raw information that drives outcomes. We strongly believe that the combined sum of these improvements cannot be underestimated. From our journey with mass dynamics so far, we've formed 4 key philosophies that will drive our path forward, where we believe that if we can solve these, we'll be well on our way to achieving the mission. Our 4 reasons why we built mass dynamics are, so number 1, mass spectrometry should be accessible to all life scientists. 2, valuable biological knowledge should no longer be underutilized. 3, to remove software as a bottleneck, this is by creating human-centric workflows, which we think is crucially important. And lastly, collaborators should get answers when they need it. So what is mass dynamics and who is behind mass dynamics? It's founded by myself, Giuseppe, Paula, and Aaron. And one of the most exciting things for me personally is the people that we've been able to bring together to tackle these challenges. I'm humbled by having an exceptionally talented, or we're humbled as a team, to have an exceptionally talented team, drawing deeply from the collective skills that we've honed in our careers with a single mission. Put simply, we exist to enable you in the audience to transform MS data to knowledge, so you can leverage mass spectrometry to drive discoveries and development better, faster, easier, without compromise. We fundamentally believe that in order to achieve this goal, we need to drastically improve access to the technology by reducing barriers to entry and create efficiencies to extract and utilize all the information that is generated, something I'm sure you'll agree has yet to mature in our field. To grow beyond the academic, mass spectrometry and proteomics requires a bit of a mindset shift. In a nutshell, we are closing the loop on the process by creating efficiencies in the flow of questions to outcomes. At a point, with enough improvements in this flow, it becomes what the overall scientific process is really trying to achieve, an iteration cycle that it maximizes efficiency where possible and can drastically improve the speed of discovery. We describe this internally as a flywheel where knowledge to outcomes gained rapidly informs on the next iteration and gets you to your ultimate goal faster. And finally, centrally to all this is the ability to collaborate throughout the approach. We put scientists in the center of our world and provide some amazing tools that make collaboration as seamless as possible. In the presentations that follow, we'll show you how the products we are developing can help you close this cycle. Rewardingly, many of our members to date have reported back on the seamlessness, the increased ability to iterate on experiments, sometimes saving many weeks to months of work. We believe in many cases we're moving users beyond or far beyond 10 times faster. As we've become a central part of some very complex and integral knowledge systems, it's important that we're clear and transparent about our principles, the areas we continually refocus on. These include being seamless, designed with ease in mind. We've orchestrated automated workflows with built integration in from day one, which helps avoid human errors and reduces the need to run manual and repeatable tasks. To be effective, we pride ourselves on quality and use techniques like benchmarking against known data sets, as well as progressive and rigorous scientific and statistical methods. We publish and we also open source. Each of these practices allows us to deliver the highest quality results. Secure. We are open and transparent about how our infrastructure is delivered. You can check out our website for more information. And because we view security as a shared responsibility, we help educate our customers about the best security practices to safeguard their own information. And lastly, collaborative. Our business models support collaboration intentionally, from shareability of experiments to keeping a large proportion of our service free. And being true to our core principle of collaboration, we've been championing the MassGeek movement for a few years now. We've also committed to continuously contributing to our open source repository at GitHub that you can check out. And lastly, creating more awareness for mass spec facilities around the world. All of these things coming together to drive mass spec awareness and grow educational assets for the mass spec community. So now it's my great pleasure to introduce Joseph Bloom first up. He's one of our data scientists who will kick off with the discovery workflow, which will be highly relevant for those in the audience that are doing mass spec-based discovery work. And for those in the audience who are running peptide mapping and quality attribute monitoring approaches, Giuseppe Infusini, our co-founder and Proteomics informatics lead, will then cover off product characterization. A reminder, if you have any questions, please pop them into the Q&A section. We'd really love to hear your thoughts and encourage an open discussion of any topics, and we'll do that at the end. Over to you, Joseph.
Thanks, Andrew. I'm so excited to be here today to talk about a discovery service. So what does discovery do? Discovery enables you to understand global protein expression changes in complex samples. Now, how did we build it? So as Andrew said, we love to work with the community. And so we've had this iterative process of feedback where you've been telling us how to build this service. Now, that helped us develop 3 key guidelines. First, experiment creation should be seamless and easy. Second, QC reports should be built into the heart of the service. And lastly, knowledge should create information. Integration of knowledge bases should help us turn not just data processing into information about protein expression, but to create real biological insights and to help you share that with your collaborators. In the next 5 minutes, I'll show you how we do each of these things. Here we see our experiment creation page. What you've told us is that experiment creation can be really difficult, involving lots of manual steps that can be error-prone that make your life harder. So we made experiment creation on Discovery really easy. You simply select your files, create your experimental design, name your experiment, and hit upload. Now you can do this with pre-processed or raw data, and and then as soon as you hit upload, it'll be a few short minutes, longer for raw data experiments, and we'll send you an email when it's done. And when you click on the link in that email, you'll come here. This is your discovery experiment, and it's got lots of features jam-packed to help you understand and explain your differential expression analysis. So we've got volcano plot, we've got your replicates analysis. You can create protein lists here and filter proteins depending on which replicates they appear in or their differential expression in different condition comparisons. We've got the QC report, which I'll talk about in a second. We've also got all your data processing results files, which you can download, your experiment design. In case you made a mistake there, you can always check it, and your workflow parameters, which enable you to see exactly every parameter we used when we ran your data, and even links to the GitHub repositories for the R packages that we're developing and using as part of that workflow. Now, we store all of this on the cloud for a minimum of 5 years, completely secure. And one of the benefits of it being in the cloud is that you can actually share that with anyone that you would like. So you simply type in an email and then select their permission. It's either read-only, means they can't create the protein lists, or full, and then you hit send. And you can share that with biologists. You can share that with the people who've acquired the data, so everyone can get on the same page when understanding that experiment. But the first thing we think you should look at when you've shared that with your collaborators is the QC report. Now, what we've heard from you is that there's 2 camps here. There's the people who are very conservative, very reasonably so, who want to go through every possible thing that could go wrong with their experiment and make sure that it hasn't gone wrong, so that when they're doing their analysis, they can do that confidently. In the other camp, we have people who try to go a bit quicker, but they do, and you always run that risk that you've missed something about your experiment, about your data acquisition that that you need to that actually makes your analysis not okay. And if you've missed that and you start doing your analysis, then that's a shame, and you pick it up later, and it's really annoying. So we thought you shouldn't have to compromise between your time and risking having to redo your analysis. You should be able to just do that quickly. So that's why we've created the QC report. So it's a series of figures that are diagnostic of different possible issues with your experiment. You can see 2 of them here. And each of them come with content that helps you understand what those figures are telling you about your experiment. And so it's very, very interpretable for everybody. On the left-hand side here, you can see the PCA plot. Now, this is great for looking at whether your samples are clustering within their groups, whether you've got an outlier sample. So this is great for understanding your experimental health. On the other hand, on the right-hand side here, you see the number of PSMs in each sample replicate. And we actually give you PSMs, peptides, modified peptides, and proteins. So you can see your identifications all the way up through the data processing. And that can be great for understanding if something's gone wrong with one of your samples, maybe data acquisition, maybe digestion or something. So that's really great for understanding if you need to rerun any of your samples. One of the best things about working with the community is you get feedback. So one of the feedback we got was one of the pieces of feedback that we got was that everyone wanted to be able to export all these figures and share them independently of the whole experiment. So the whole QC report is exportable as a PDF, and also every single figure there is exportable for you to share with your collaborators. Now, once you've got that confidence in your experiment, which can happen quite quickly, you'll be ready to analyze your experiment. Now we have several layers of analysis here that integrate using information from UniProt, from gene ontologies, from Reactome, all centered around turning data into real knowledge. Now, what we heard from our users was that from you is that experiment processing. You want to be thorough about it, and you want to make sure that you get as much real biological insight as possible out of looking at which your protein expression. But you also, you hear about people having 100 tabs open, that you have to go everywhere to try to bring all this information together to make it possible to get those insights. And so what we set out to do was not just make it automatic, not just make it easier, but to really bring all of those insights from across the web and all those bioinformatic resources that people work so hard to develop and help you access them better. So you can do differential expression analysis. We use Lima to do that. You can do overrepresentation analysis with those protein lists that you've curated, which I mentioned earlier. But you can also do enrichment, and this is a beta feature that I'd love to share with you because we're really excited about it. Now, the way it works is we use camera to look at the ranks of all the proteins that have been expressed in each of your comparisons. And then we look at groups of those proteins and how highly they're ranked. And those groups can often correspond to real biological concepts, such as biological processes, for example, or reactome pathways. And that can find signal in data that's otherwise quite messy, and sometimes that can be really interpretable really fast. So we did that with a data set that we love, the HER2 data set, and we got some awesome results. And you can read about these more in our app note, but I'll tell you a bit about them. So it's the study that you see on the left here is a study looking at the evolution of breast cancer therapy resistance. So these breast cancer cells, some of them develop this epithelial to mesenchymal transition. They're then resistant to the HER2 targeted therapy. And obviously we want our treatments to work, and we want to be able to get all of that cancer. So we want to understand why they're developing this resistance. Now, the data from this study wasn't ever linked to this SLIT-ROBO pathway in the original paper. But when we analyzed it, we saw that these pathways were enriched in the Reactome enrichment results coming from camera. Of course, this was after we were confident in the results, after we'd done the QC report and looked through all those figures. So we had that confidence. We then did the analysis, and we saw that they were related. We then looked into that a little bit more, and we found that there were other researchers who were talking about this, who discovered this, but that wasn't brought up in this paper. So that was just so exciting to see that you can bring out real insights that you can validate by looking at other resources, But maybe maybe you'll be the first one if you can perform that analysis. So we were lucky we could confirm this one, which was really awesome to see that this may have been able to accelerate that research. So you can read more about that in the app note on our website, as I've just said. Now, that brings me to the end of this section. I hope I've explained well enough that discovery is all about making your life easier and making your experiment process faster. So easy experiment upload, credibility around your data, understanding it, moving forward with confidence, and then relating your insights to real biological concepts that are inherently more publishable, more shareable. Now we've got that Journal of Proteome Research logo there. I'm really glad to say this. We've worked quite hard, and we've been told recently that short of a few minor revisions, we'll be publishing the Discovery Service in JPR coming soon. So that was really awesome to see, and you can read all about all our methods there and how we've validated everything, and it's just a great process with JPR. I'll now pass to Pepe to talk about protein characterization.
Thank you, Joseph, for taking us through discovery. I'm actually really excited to finally surface the work that we've been doing and how we can support our members doing the protein characterization work. And I don't have to tell you that protein characterization plays a very critical role in the discovery, development, and manufacture of products that are produced relying on complex biological systems, where the inherent variability of the systems makes a thorough characterization and monitoring of the product a necessity from development down to production. So while there are multiple methodologies to approach it, a common refrain that we heard from our members is that this invariably becomes a time-consuming and labor-intense task, even for highly qualified analysts. So we set out to build a solution that can help ease this pain and recuperate this time. Now, the first two approaches that we tackled are peptide mapping for the characterization of protein primary structures and PTMs, and high throughput attribute monitoring to accurately quantify product quality attributes in large batches. So first, let's talk about peptide mapping. And in reference to the core parts of our service, today I'll touch on how we can enter the data so it is an easy and simple upload and processing step, how to access the quality of your experiment and navigate through the identifications with interactive visuals, and lastly, how the identified PQAs build the foundation of the product knowledge and feeds into multiple attribute monitoring. Now, first, this isn't too different from discovery. What you see here is where you can create a new experiment and define the key parameters for your search. And in this space, you can build company-wide information like customized PTMs, custom glycan databases, FASTA databases, that you need to add to the search, and all is saved and ready to be used and reused as needed. Now, also because we're leveraging cloud infrastructure, you're not bound to a single or even a few seats on a computer, and you can have any number of experiments submitted by any number of users simultaneously, and all the results are seamlessly viewed and shared within your organization. So one thing that we heard from our members is that one of the big challenges is that is the large amount of mass spec data that they need to sift through, and it is really hard to navigate it all. So we made the inspection of the data more visual to aid the analysts navigate the results. And what you can see here is a representation of the protein sequence and the resulting coverage. And of course, this is usual. It can give you a summary of your experiment results and all the identifications, and all the modifications that you have identified. But we made this visualization interactive so that you can hone in specific regions of the protein by selecting a peptide or a region, or indeed a specific modification to be sent directly to the relevant evidence underlying the identification so that you can review it. So what you can see here is a simple example of this review window for a fragmentation spectrum, where on the left, there is a list of all the identified peptides and PTMs in the protein region of interest. In the center, there is displayed the best identification for the sequence and modification combination. And on the right-hand side, there is a list of all the PSMs that yield the same identification. We made it a little easier to compare fragmentation patterns of sequences carrying a modification by automatically fetching the fragmentation spectrum of the unmodified species, so that you can easily spot expected or indeed unexpected patterns in the data. But of course, MS2 spectra is only one part of the story. And so we also provide other visualization to gauge the validity of the identification and the quality of the MS1 feature. So, for example, we show you the three-dimensional plot of the MS1 feature from which the PSM originates, and the two-dimensional chromatogram view of all the isotopes that we detect, and always trying to facilitate the comparison between the modified and the unmodified from species from your peptide sequence. So in here, I'm simply showing an identification of glycosylated peptide just to make a little bit of a point, and that at least in ideal conditions, such as the one that shows here, we can highlight the presence of oxonium ion or peptide backbone fragmentations, and fragmentations within the glycosylation. Again, what we heard from our members is that screening PTMs identification is really only one part of their task, and that compiling the data for to quantify the relevant peptides and communicate the results, it's a laborious, time-consuming, and often tedious work. So we have built the service to allow you to immediately move from identification, screening to the quantifying the peptides and PTM without wasting time collecting the data. And we immediately provide relevant visualizations, and the resulting analysis can then be easily shared with your collaborators or clients directly in the app. Now, by doing this, you will also be building within the app the knowledge about your product and of the important PQAs that should be monitored in later stages of the development process. And this knowledge is then also automatically transferred to our other high-throughput monitoring service. So this concludes the brief section about the peptide mapping service, which in summary, it allows you to analyze experiments seamlessly and at scale, and to scale with ease thanks to the cloud infrastructure. We built interactive visualization to help you navigate and assess the evidence in your experiment, and it will allow you to build organization-wide knowledge that can then be easily accessed, shared, and ported into high-throughput RGB monitoring. So let's move into the other type of experiment that you can analyze in mass dynamics, and it's multiple attribute monitoring. With the evolution in the regulatory landscape to promote quality by design, high-resolution mass spectrometry is quickly becoming one of the most prevalent analytical tools, particularly because it allows the analysis and monitoring of multiple PQAs or CQAs simultaneously. Now, as I've been saying and mentioning throughout my talk and the presentation, we heard from our users that there are a lot of time-consuming steps in the current analysis pipelines. And so we endeavored to build a tool that could alleviate and reduce significantly the amount of time spent in repetitive tasks, and we have automated parts of the analysis. Now, attribute monitoring is probably the most mature application we have, and it has some really rich and in-depth features. So I'll try to give it justice. While I'm going to focus on how you can gain confidence on the results of an experiment through automated quality checks, how we present the data to easily perform quantitative analysis, and how you can record insights, share, and collaborate with your clients directly in the app. So first of all, I should mention that the processing in multiple attribute monitoring, our multiple attribute monitoring service is solely based on MS1 data and three-dimensional feature detection rather than extraterritorial chromatogram. And this allows us to perform very accurate quantification of the relevant PQAs, even in short gradients, maintaining a high degrees of specificity. But it does require in-depth quality checks to assess the health of the overall experiment and of your LC-MS system. So in the first instance, we perform a handful of automated tests, like like negative control to assess the extent of carryover, identity peptides, just to ensure that you're actually analyzing the correct product, and suitability checks where we monitor a standard sample against several user-defined parameters. Like, for example, here you can see a check for sensitivity where you would expect a specific set of peptides to be detected above a threshold. Now, both the standard, the criteria, and the thresholds are all user-defined, and you can add as many as you see fit. But the point is that upon completion of the data processing, the first day you will see, you will be provided with a summary of the results to give you confidence in the status of your experiment. We also provide more QC visualization to assess the status of your system, or if something goes wrong in the system suitability checks, to diagnose possible issues. Also, historical information about your system can be stored and can be easily viewed and to assess the performance of your instrument in time is to spot trends or biases in your data. We heard from a member that assuring the quality and collect the relevant data to perform quantitative analysis is a huge bottleneck, and they wanted a higher throughput on their instruments and faster data analysis cycle without compromising in quality and without increasing headcount. So the question is, how do you run more samples and more replicate without increasing the work for the analyst? And the approach to the problem that we took was to use an exception-based review. So the application performs all the quantitative analysis based on user-defined patterns and requirements, presenting visual like the one that is displayed here. And here, for example, you can see on the slide before, sorry, what you can see is the comparison of varying composition of 4 glycoforms between 3 different samples. But when the workflow spots an anomaly in the data, we flag relevant samples to your attention, and from there you can easily access the underlying data to take action, such as, for example, removing a bad replica or a feature that has some unexpected interference. So when you dig into the data, we present you with different views to assess the quality of the detection. And here, just as an example, I'm showing you the results of the three-dimensional feature detection, where we can easily differentiate, or you can, in that case, very clearly see the difference between the MS1 feature to be targeted and to be quantified and unwanted interference. Now, using three-dimensional feature detection allows us to gain better specificity with respect to XSE, and this results also in reducing the amount of manual intervention in the extraction parameters and reducing the time to complete the analysis. The next bottleneck that surfaced when we spoke to our members was that collecting the results and communicate them to their collaborators or clients, right? So we built the ability for you to generate different type of plots directly in the app in order to gain better insights about the data. And what you see here is the analysis create page, where you'll be able to create 3 different type of plots, which will allow you to subset and slice the data as you need. So these are a couple of examples of plots that you can generate in the app, and this section is designed essentially to allow you to build the insights about the product and to just extract information from the data. But our members didn't want to waste precious time compiling all the work that they've done into reports and share the insights to the stakeholder. So the app, first of all, automatically generates the report, which will have a record of everything that has happened in the app, including any data review steps you might have taken, any plots and insight that you might have recorded in the app. And of course, you can download the PDF and use this as a record in your ELN. But we never viewed the report to be used as the primary communication tool. Our idea and what we built is a mechanism to share the results directly in the app, so that your clients or collaborators can interact directly with the graphics. And so within the application, we allow you to generate the plots and the graphics to communicate the results. And in a matter of speaking, you arrive to a point that you can publish them, and share them with your client and collaborator. And on the client side, they can view these interactive plots you generated and read any comments and insight that you might have recorded to enhance and speed up this conversation and communication. So, removing essentially the need of compiling lengthy PowerPoint presentation, email conversation, and wasting time in lengthy meeting. We arrived at the conclusion of this section about high throughput I should be monitoring, and when I hope I covered, how you can be confident on the quality of your experiment and instrument setup via automated system check, how we can speed up the quantitative analysis via exception-based review, and ready-made visuals, and how you can get insights into data and share and collaborate with your colleagues directly within the app. So thank you very much for your attention, and over to you, Andrew.
Thanks, Giuseppe. That was great. Thank you to both you and Joseph for giving such a wonderful overview of the products as they are. So where to now? I've been more vocal of late about how proteomics, I think, is really becoming the final frontier of medical research, and it's for really good reason. I truly believe that we will accelerate better treatments, diagnoses, and preventions once we better understand the proteome. There has to be a significant change in the way we move from discovery and through the entire product development cycle. And so it's no surprise that Mass Dynamics is going to be running initiatives to continue to improve upon making our approaches even better, faster, and easier to help you and many more life scientists move as quickly as possible through the life cycle of processing data, assessing quality, leveraging even more available global knowledge, and enabling researchers to take action and continuously experiment, all without the compromise of security, effectiveness, and seamlessness, and of course, with a strong collaborative focus. So that's where we're going next, but you may be excited to hear what's new now. On behalf of our incredibly hardworking team, I'm excited to announce that we have just last night released peptide mapping to production. So now, once you sign up and get your free membership, you can run a demo data study. This is based on the NIST monoclonal antibody reference material, and check out our peptide mapping features from your very own living room. Over to you, Paula.
Thanks, Andrew, Joseph, and Giuseppe. That was fantastic. Look, in closing for the presentation, we would really like to take this opportunity to acknowledge all of the members and community who have helped us so far. So whether that be from testing out our new features, sharing their thoughts on the future of mass spectrometry, referring our service to collaborators, or collaborating with us on feedback and requests, it's all been incredibly appreciated, and we couldn't have got to where we've got to with our service without their help. And so finally, if you believe in this mission, you can help too. So what you can do is jump straight into app.massdynamics.com and get your free membership. So this is going to give you access to our free services, including Discovery, which Joseph went through, and now the peptide mapping service, which supports the processing of demo data that Andrew just announced, and that's based on a published study. So from there, you can share feedback, suggestions, and of course, share your experiments. And if you love what you see and you want to know more, you can absolutely book a demo to find out how to upgrade to a paid plan or to get a little bit more in-depth information about the services. And if coding or reading is more your thing, you can certainly check out our GitHub repository, or we've got our app notes, tech notes, and our papers on our website. So in closing, in case it's not come across in this presentation, we absolutely encourage engagement from the community. And with that, I'd like to open up Q&A. If we could welcome back the speakers on stage. So there's the four of us here. Now, while they're making their way up to stage, you should be able to see the link pop up in the Q&A section. So app.massdynamics.com. And yeah, would love if you could jump on there, give us some feedback about what you think. Now, to kick off Q&A, we've got some questions come through, so thank you so much. And please, if you're holding off, please pop it in there. It's really valuable for us to have the discussion now. Now, the first question I wanted to put up was from Anonymous. Now, So the question is, do you support protein peptide table outputs from any software to run the discovery workflow? Giuseppe, would you like to address that?
Yes.
Joseph.
You can go.
Okay. So currently we support MaxQuant preprocessed data for TMT and LFQ experiments, but we've also got a generic format for preprocessed data. So really any software that you process, you know, a lot of them will produce this wide form protein expression output, and we've got this generic upload format, which is very easy to get any sort of table into that format, and then you can do your protein level differential expression analysis. Peptide level for peptide level data, we don't currently have that, but it's something that we're certainly listening out and see if that's a priority. We can certainly make that happen. You know, balancing it with all the other requests that are coming up.
Yeah, absolutely. So certainly, shoot us a note. We've noted that down already as some great feedback. So thank you very much. Thank you, Joseph. Now we have question 2, and it's also from Anonymous. Do you handle mass spec data from all vendors? Giuseppe, would you like to address that?
Not at the moment. Right now, for the raw data, we handle just thermal instruments or orbiters. But you know, we'll be worked tirelessly to include as many mass spec that we can in time, right?
Yeah, fantastic. And it's worth mentioning as well, with Discovery obviously being generic format, there is certainly that direction that we're going in terms of being able to handle more and more formats. Thank you, Giuseppe. Now we've got question 3 also from Anonymous. Should we use your platforms to just acknowledge the use of platforms? I'll pop it up there, it might be a bit easier. So do we acknowledge the use of platforms like using Skyline, or will you be included in the author list, etc.? So Giuseppe, you could possibly talk to this directly about the most recent paper that was produced and how we handled that.
It's just a software that you use. It's not. You just acknowledge and reference either the paper or the service that you use. But the data is yours, the work is yours. We're nowhere in the paper. We'll do our own publications about the software so that you can confidently use it and easily reference it in your paper. So that's our aim, at least.
Yeah, absolutely. And I think to add to that as well, the most recent paper that's come out where we've been cited in the methods and materials, there was the ability to put a public link in the paper as well. So basically people could read that author's paper and then they could click directly into the interactive data. So that was super exciting for that particular author to be able to do that in their paper.
I think that's a really good point. Sorry, Joseph. I think it's a really good point, Paula.
Yeah, I was just going to add, speaking of references, one of the things we've been doing in the app is to make sure that every feature, actually, if there are any extra references that you want to put in there, say you've used the enrichment feature or the overrepresentation analysis feature, we'll put in the references that you need to make there, so you can have everything to pair in your paper to make sure you've got all your citations.
Yeah. Fantastic. Okay, so there's one more that's coming through. Please keep them coming. This is fantastic. We've still got 15 minutes of time here. And it's from Anonymous. So no WIF files, WIFF files from triple TOF science, question mark? That's a new feature request, Pepe. There you go.
No, it's not an instrument. Yeah, not yet. We're working to get generally TOF data into the app. We've been driven very much by our users, the need of our users. So we were a small team, we're a young company, so we're just coming out. So we are building things as fast as possible, and we give priority to the things that that we definitely need, that our users need. So it comes down to getting on board and let us know what are your priorities, what are your needs, and so we'll stack them up to get to it. And if if SciEx is the next thing that we're going to do, it will be, of course, we're aiming to do as much as possible, both in workflow and in type of analysis, or different differential expression, different instrument, and put it there, DIA, TTA, and things like that. So that's our goal. The question is what we will do first. It will probably be determined by the people that are onboarding the app and letting us know.
So just to add to that, I think with discovery workflow, that by pre-processing your data in some other software, you can import the protein tables in so that you can actually do and use the triple TOF SciEx. So I just wanted to add that, Giuseppe.
Yeah, no, that's fine. It's true. I kind of forget that part of it.
Oh, fantastic. Just see if there are any ones that pop up. I mean, I think it's worth mentioning, there seems to be lots of question about data, which is absolutely wonderful. It's worth mentioning the demo data that we're using for our services really quickly while we wait for the next question. So Joseph mentioned the HER2 data set that we're using for discovery, which we absolutely love, and we continue to acknowledge the authors on their wonderful work there. You would have seen in some of the slides, which is Giuseppe's slide, we're using a study data based on that we got out of Pride, and that's based on a NIST monoclonal antibody. And we're really excited as well to we've been partnering with NEBIT on a third data set that we've been using. So we're really excited. So please stay tuned for that. Some really exciting insights that have come out of that. And I mean, with that, you will see as well, we didn't cover it, but on our discovery service, you can actually see public links or public studies in there. So if you do have a data set that you absolutely love, or a paper that you just adore and continuously cite, we would love to hear from you, because we can certainly process that and put that in that same area, so then it can be publicly accessible and interactively worked on by people around the world. So please do reach out if you have any ideas like that too. Okay, there's another question that's come in. It's another SciEx question. So can we convert the .wif files from SciEx to MZML files and use them?
Not yet either. The question is not really about the converting one into the other. It's more about algorithms behind. At the moment, the ones that we have are very tailored to high resolution Orbishrap data. We're moving to port and to expand that into using different type of data, like TOF data. So as soon as we can do that, that won't it will be either WIFs or MZMLs, depending on what we can do. But it's more about the algorithm behind and the instruments. Yeah.
Fantastic. Thank you. So we've got another 10 minutes. We'll see if another couple of questions come in. But I've got a question that, I mean, over the last few years, we've talked to lots of different people in the community. And I think one thing that's come up a fair bit is around this whole concept of, black boxes and what's in there. So, Andrew, if you wouldn't mind addressing perhaps how we're thinking about that.
Yeah, no, that's a really good point. I mean, it's the same, I wouldn't say criticism, but I say concern from a lot of the experts in the community that they don't want to move away from having a handle on the data. They don't want to move towards having these black boxes where they don't quite understand what's going on inside. And I think our broader philosophy is that we're not building a black box, but we're building a transparent box. I think the important thing is that there is a box that's helping to automate and to move people much faster through the sort of traditional and very manual processes towards the analysis goals. But that transparent box, I think, is really made up of it's good, high quality, open source code that's transparent to the community. So we are working with the open source community. We use open source tools in our workflow that other people have created, and we fully acknowledge them and we contribute to them. And we will maintain, for the bulk of our statistical and analysis pipelines, we will maintain at the core, I think, an open source philosophy on this. I think this is hugely important going forward. I think it's important for us. It's important for us to be successful going forward, because I think we need to work as a community to solidify what are the major and most rigorous ways of analyzing data sets.
Yeah, I'll just add to that, Andrew. Like when we went through the process of publishing or submitting the discovery paper, you know, it forced us and helped us look at how we really show as much of that as possible and become as transparent as possible. And I think that was a really sort of conducive experience. So, you know, I can't wait for us to do that with more papers and more parts of our service, so that everything's out there, everything's completely sort of open source, published, and really, really understandable by the community.
Yeah, fantastic. Thank you, Joseph. Now, we've had two questions that have popped up, and they're related, so we can certainly address them again. So one of them is, can you talk more about your data security again? And then secondly, would you be able to comment on compliance? So I can take this question. So we did touch on it. It's certainly one of our major principles, being that we are secure. So what I will start by saying is that if you hop onto our website, there is an entire page that is available, and it talks about compliance and security. So it goes into detail really openly and transparently about how we're addressing each section. So things from privacy, things from the infrastructure that it's built upon, how we handle data, and things like that. It states it really specifically. Now, the reason we've done that is because we do know that that members and clients have different requirements. So it really is dependent on what you need and to what level. So what we suggest is, if you have, if you want to talk in more detail, we would love to talk to, say, your IT teams, for example, to see what their particular requirements are internally. Now, we do work with biotechs at the moment, and we have gone through rigorous tests through their processes, and that's certainly, you know, it's given us that unbelievable foundation of security and compliance as a company. So I guess the bottom line is to please to get in touch, have a look at that detail on the website. It is incredibly extensive, and do let us know if there's anything missing that you require. Now, there's another question that's come through. Now, did you say, is this too good to believe, did you say your platform is free? Now, this is a really great question to address, and I'll start, and if anyone wants to chime in, they certainly can. We believe that there are base level services that the entire community needs, and to that end, we've exposed things on our service in the app.massdynamics.com environment that are free. So things like the differential expression analysis, each of the features that Joseph talked about. Now, when it comes to the paid plans, there is a bit of a shift here, and it goes into the realms of things like the extensive raw data processing, for example. It goes to things like having premium support. It goes to things like virtual private cloud infrastructure, for example, and custom domains and services like that, and not service features like that. So yes, there is a base level free plan that we will always keep free, but certainly as you build upon the features that you need, that's where we, you know, that's where our commercial model kicks in. And I want to point out really specifically as well, I mean, this is how we're going to build a sustainable business, by having, yes, we need to make some money to keep this sustainable, but we certainly have a very much a community bent here to be able to support that. So I hope that addresses that question. And please get in touch if it hasn't. I'll just see if there's any more that come up. These have been absolutely wonderful, everyone. We appreciate it. We do know that people have Zoom and online fatigue, so we're very thankful for your engagement. I'll give it a quick moment. And look, there's a bit of free space here. I'm going to put it out there. I'm not sure if you guys saw the hashtag MassGeek, but if you have any thoughts on that, we'd love to hear it. We quite often get asked, you know, how do I get onto MassGeek to be a MassGeek? We would love to hear about your MassGeek stories. So very, very quickly, pure plug on this, but I personally love it as an initiative. We asked 3 questions about 3 moments of, you know, how you fell in love with mass spec, where you see what that magic moment's going to be for mass spec, and the most proud moment of your mass spec career. If you're able to answer those questions, we would love to hear from you. And then we're happy to push those answers out to the community and really keep building upon that, you know, really pushing the good word on the amazingness of mass spectrometry. So please get in touch about MassGeek moments. Okay, so we've had one more question. What does 10x faster? Now, I'm going to assume that sort of means what do you mean by 10x faster? So Andrew, it might be worth addressing the concept you talked about in that slide a little bit more about how we're closing that loop faster, and
yeah, I mean,
sometimes they customers can't get to the end, can't get to the end of the cycle.
No, yeah, I was just thinking. I think you're probably thinking the same thing, but probably one of the best ways of explaining this is, talking about some of the things we've helped our customers achieve, and some of the things that we've spoken to other people that are yet to be customers but have some significant challenges. So, it's not just the processing. It's not processing, the search data faster or things like that, but it's actually what you do with it. And so it's really providing, the links between the different disparate groups and allowing them to seamlessly work together. That provides, we've found in quite often, it's more than 10 times faster. So instead of taking, months to do an experiment that has literally been sort of maybe one person is really taking hold of, because you've given them an environment to share and talk to and collaborate on data, and given them the tools to link from one step to the next, that it creates these enormous acceleration of how do you get to insights within data. And I think this is something we're going to continue to hone. I don't think we've got it completely perfect yet, but I think over time this will become even faster. I think it's sort of something I think you almost have to experience as well. It's once you upload a data set that you can almost instantaneously start to look at networks and go enrichment analysis and these things. It's something that would normally take days, quite often. And I think one of the biggest things that happens when you're analyzing data sets, and it certainly used to happen to me a lot, where you would set up something, you would generate something to generate a result, but because it took a day, you'd almost kind of forget where you left off, and it was always a bit slow to pick up. But because it's there and it's immediate and it's completely iterative, this is where these huge speed ups are. It keeps it in the forefront of our minds, and this is where we can make quick, fast decisions. And added to that is this layer of collaboration that can now happen, which means that it's, when you generally, a lot of these delays come from, maybe emailing somebody or contacting somebody, and they take time to respond because they're busy. But when it happens in app, everything just moves so much faster. I don't know if you want to add anything to that, Paul.
Probably the only thing I'll add is probably a small anecdote that sticks in my mind really heavily. So I recall one of the meetings we had where we were doing some user testing, and we had an MS expert sitting right next to, like literally in the same room. This is possibly pre-COVID, and they were sitting and interacting on the same service. They were talking the same language. They were communicating just so efficiently. And for me, it was a huge defining moment, saying, "Wow, we can really close that loop close that cycle by getting biologists and mass spec experts on the same page really quickly." And that was like I said, incredibly proud and awesome moment to see. Now, a question has come up around, can we use peptide mapping now? So the answer is you can use it with demo data. So if you hop on to app.massdynamics.com and you create an experiment, you'll be able to see a big button that basically says use demo data, and you can do that. Beyond that, it'll take some time. There would be I believe there's going to be a couple of free raw processing experiments within the free plan, which will be released very, very soon. So yes, the answer is yes, but it'll be demo data. So like Andrew said, from your living room or from your, from your wherever you like in your house, if you happen to be in lockdown right now, you can have a go at it. You don't need to go into the lab or anything like that. You can certainly use it and try it out. Just see if there's any other ones that come through. We've got 1 minute to go, and we do want to get people off and running. Some people are possibly going into the evening and looking forward to some good rest. Okay, there's one more question that's come in, so we'll cover this one off, and I'll pop this one up. This is a great one. So are there limitations on experiment sizes for the free service? This is a wonderful question. Giuseppe or Joseph, would you like to address that?
Joseph, you're going. I can see
yeah,
it. I can say.
I don't think so. I mean, no. Yeah. I mean, we'll see what you upload, I guess, but probably not.
We haven't broken it yet.
Structure is designed to scale. Yeah. So.
Yeah, we don't have any hard limits. I guess I think we have experimented with the big experiment. The thing tries us out. I guess that's the question. Let's see what can happen.
Yeah, we've
I think what they're saying is Sorry, go Joseph.
I was going to say we've certainly had some very, very large experiments.
Yes.
That's for sure.
Yeah, we certainly have. I mean, from perspective, I think they're talking about like for the free service in particular. So no, there are no limitations in terms of what you can upload. So you can have as many conditions, as many replicates, as many files and runs as you like. There is no limitation there. And I think what Giuseppe and Joseph is saying is bring it on. let's see. Let's do that. All right, so look, we're just ticked over 8 o'clock, so I'm going to wrap it up there. It's been an absolute pleasure delivering this webinar to you. Thank you so much for coming. We've been Mass Dynamics and we've taken you through, Andrew talked us so wonderfully, talked us through the bottlenecks that we're facing. Joseph took us through our differential expression discovery service. Giuseppe talked us through product characterization and how that's speeding things up in that space. And then we've gone through these wonderful Q&As. Thank you so much for putting them up and opening up that discussion. We look forward to seeing how we can help you and your collaborators transform your mass spec data to knowledge. Better, faster and easier than ever before. So please be well and we hope to be processing your experiments really soon. Thank you.